it's Emily with the Broken Science Initiative, and in this video, we're going to delve into the intricacies in the world of randomized clinical trials, which are often referred to as RCTs. And then in a companion video, I'll get into more detail about drug trial phases, which are the second part of this. So these trials are basically the backbone of what is often referred to as evidence-based medicine. And they really are the guiding principles or decisions that we make in terms of deciding what treatment to recommend or to receive. So in this sort of video, we're going to get into the exploration of the RCTs and analyze some real-world examples to comprehend their significance in medicine, as well as some of their shortcomings. So step one is study design and hypothesis. And I think let's just sort of start with this is the foundational phase of an RCT, which is really the study design and hypothesis formulation. So researchers begin by crafting a hypothesis that aims to answer a pressing medical question. So an example of this would be to imagine that there's a study of looking at sort of like novel antiviral drugs, and they're going to compare that to a widespread viral infection. So the hypothesis on hand is going to be something about whether this new drug surpasses the current treatments, the standard of care, in reducing a viral load. Step two is patient randomization and treatment groups. In this stage, the spotlight is on patient recruitment and random assignment to treatment groups. So the essence here is really in the randomness, and the idea is to eliminate bias by ensuring that there's sort of an impartial evaluation. So picture hundreds of patients are enrolling in a study. Half of them are assigned to a treatment group and are receiving this new antiviral drug, while the other half is comprised of a control group, which is sort of like the placebo group, which is maintaining the existing treatment. So we're comparing those two. Step three is the data collection and blinding. So this stage really emphasizes what should be a meticulous collection of data, and a crucial element of this is blinding. So data is systematically gathered under various parameters, such as viral load, side effect, and patient outcomes. And blinding safeguards against bias is the hope that might affect the treatment and affect our ability to sort of measure the effectiveness. So let's say consider sort of researchers are diligently trying to gather what the viral load is while also monitoring other symptoms and documenting any adverse effects that might come about. Maintaining a double-blinded study is meant to ensure impartiality. The fourth stage is the statistical interpretation and testing, and this is dedicated to the realm of statistical analysis and data interpretation. So researchers employ statistical methods to determine whether the new treatment demonstrates a superior performance compared to the standard treatment. And this is done with things like p-values. So check out that video that we've done on p-values. So researchers will analyze, and they may find that patients in the treatment group exhibit a significantly, which is p-values, greater reduction in viral load compared to the control group. This statistical significance points towards, theoretically, the potential of this drug being superior to what the current treatments are on the market. Step five is when we move into the real-world application and sort of publication. So this is about disseminating the results that have been found in the trial, and they do that through peer-reviewed scientific journals. Now, people think this is a crucial step because it's really bringing the information of what we found in the experiment to the broader medical community and asking for feedback and, in an ideal world, having people explain why we didn't do the study right. That doesn't happen as much as we might like it to, and that's highly problematic. We have a lot of information on problems with peer review, which I highly suggest that you take a look at because it's not a gold standard as much as everybody wants it to be. It's very reliant on p-values, which we don't believe are any point of validation or indication that the study can be replicated. And you add to that the sort of idea of consensus thinking around peer review, and it becomes pretty problematic. So we actually have on Broken Science most of the leading high impact journal editors calling foul on their own industry, despite the fact that the mainstream media doesn't seem to really cover this. And they take anything in peer review and they accept it as having been fully fact checked. It is not. So check out a video on peer review too if you want more information on problems with peer review, which are many. But this is an important stage if we're looking at randomized controlled trials as a way of, sort of deciding new treatments, drugs, interventions, and how do we measure them compared to what we already have in the marketplace or even no care at all. And so this stage is considered to be very important because it's sort of a consensus agreement that this treatment and this experiment has been done properly. And let's talk about some of the other limitations. It's really important to acknowledge that RCTs are better than observational studies, which we have videos on, but they're not, proof of something per se, right? They're indicative of something interesting. They found something. And I would argue that unless it's replicated, it's not really as meaningful as people want it to be, but it is what is generally used as moving the needle forward in terms of getting drugs approved and, us sort of accepting the treatment as a valid form of dealing with the underlying condition that we're hoping to treat. So another limitation that we see in RCTs is that there's a sort of tendency to have homogeneous study populations. So they're not necessarily representing a wide, diverse range of people in the real healthcare world. We see this as hugely problematic when you're looking at women and female bodies, which have been underrepresented in clinical trials because in the 1977, Congress banned women from being allowed to be studied in clinical trials if they were of childbearing age. Now that's been reversed, but it's still wildly disparate in terms of women being represented equally to men. Women's bodies are totally different than men's, and we experience different diseases and we have different symptoms. So you want to be careful to look at any kind of randomized controlled trial that's being recommended to you. If you're a woman, it'd be great to know that there were women in that study and some representative population. And if you can dive in a little bit deeper and see how if those women had different side effects or efficacy of the treatment was different for women than men, it's actually oftentimes more than people probably like to acknowledge. There is a big difference with sex differences. So that's something that I would encourage anybody who's looking at this from a patient perspective to dive in on a little bit. Randomized controlled trials are all, because they're published, you can look at them. It's also really important to understand why clinical trials fail sometimes. And oftentimes pharmaceutical companies will try the trial again. So it's always sort of interesting to go back and look and see, did it fail before and why? And then what did they change in order to retry or redo that sort of specific trial that you might be interested in? So one of the first things that always comes up is just that there was an inadequate patient enrollment. So they didn't get enough people. So the enrollment process to get participants on board, maybe they started and people dropped out, or they just didn't have enough people. And so the statistical power of the trial would make it difficult to draw any meaningful conclusions. We also have things like adverse events. So morally, if something comes up as a severe side effect or some adverse event, which can emerge during a trial, then the study it has to be discontinued at that time. That's really important to know about in an earlier trial, because sometimes things can be slightly modified so that isn't presented, but it might still be a risk factor that you want to know that an earlier form of this drug, for whatever reason, impacted the subjects in a negative way. And so you also, if you're really going to dive in, you kind of want to know, did they change anything about who the subjects were, or look at a different cohort of people that they're studying. And if you represent the prior group, that's probably important to know because it's certainly a risk factor. There's also the ineffective treatment option, right? Where they test it and they find out it doesn't actually work. It's not that effective. And so the standard of care becomes the default and it doesn't move farther along in the process. So there's also this idea of non-compliance, which is where, let's say you have the subject population doesn't really adhere to what they're supposed to be doing in the trial, and that prevents the ability for being able to study it. So people drop out, or you realize that they're not really taking the medicine, or there's some other part of this intervention that they're not compliant with. And if you find that, then obviously you're not able to really test that group. So that would also cancel the trial. And then there's like this idea of statistical noise, which is really important in some ways, especially because we're such critics of p-values. There's some variable or confounding variable that's clearly weighing on the subject group or the comparison between the two that makes it really hard to say conclusively that the drug is effective or the treatment is effective. And in a highly ethical environment, that should cancel the trial, because it's saying Even though we've studied this, there are too many other things going on, and we can't conclusively say anything about the effectiveness of this treatment. Now, unfortunately, that doesn't happen as often as we would like. And sometimes you look back at these trials and you think like, holy smokes, there was a lot going on here. This probably should not have moved forward, even though it did. So that's another one to sort of look for. And that goes into p-hacking and a lot about how p-values are measured or changed. That, again, I would encourage you to watch that p-value video that we have. So after the sort of successful completion of a randomized trial, then the journey of this intervention continues on to the FDA drug approval process here in the United States. And this process involves sort of several key steps, which I'll get into more detail about in the next video. I think understanding RCTs is really important in terms of understanding also how they move into this drug phase sort of part of the trial, because that's when they get to bring it to market. And there's certain requirements to bring it to market. And then there's actually continued testing that's supposed to happen after it's brought to market, so that when it's, introduced to the larger population, we can keep studying with, obviously not just test subjects, but now real people who are taking it, who we can learn a lot from. And we should be refining those processes as we go. I hope you'll watch that next video too.